Welcome, everybody. Thank you very much for your interest in our work. PlasmoFAB, a benchmark to foster machine learning for Plasmodium falciparum protein antigen candidate prediction. First of all, I wanted to tell you a little bit more about malaria than you probably all know. Here are some facts from the WHO from 2021. Nearly half of the world's population was really at risk in that year of getting malaria, and about 247 million cases of malaria were estimated in that year worldwide. It is estimated that a number of malaria deaths was at about 619,000 people in that year worldwide. And the WHO African region is most affected by this. So 95% of malaria cases and also 96% of malaria deaths are in that WHO African region. And children under the age 5 are disproportionately affected by the disease. So they account for about 80% of all malaria deaths in the WHO African region. And how is malaria caused? So it's basically caused by a parasite, and that's a Plasmodium. There are several Plasmodiums which can cause malaria, but the most important one is Plasmodium falciparum. And you can see the complex life cycle here. What you can also see is that there is a vector involved here. So in this case, a mosquito, because the disease is not transmitted from human to human directly, but through those mosquitoes, because they bite infected humans, take up the Plasmodium, and then bring it into another human being. And in that course of this life cycle, new human beings are infected. So there is a treatment that's available, and there are several treatments available. So you probably all know about the Nobel Prize in 2015 to UU, but diagnosis is not easy in those affected areas, and also resistant variants are emerging, and also resistant against those insecticides which are sprayed onto those bed nets. So if you really want to eradicate malaria from a whole country, then we're still having a lot of problems to really get this efficient. And there are certain challenges there if you want to develop a malaria vaccine. So you might say, okay, yeah, just get a vaccine and we're done with it. But okay, for other diseases, we've also seen that this can take a little bit longer. But for malaria, also, it took a while until it was possible to get a vaccine. And this is also the case because there are many, many proteins for Plasmodium falciparum. So if you want to train the immune system to recognize something from that parasite, then you have to really select what to show. And this is not easy because of that large variety of proteins, and also due to the large genetic diversity of those different proteins there. And there are also various immune escape mechanisms. So there is a vaccine, but the efficacy is just at 75%. And there's no steroid protection. So meaning that those people who are vaccinated can still get the disease and can still transmit the parasite to other humans through that vector process that we just talked about. Okay, and therefore we thought that we could assist that process of getting a better vaccine by our computational methods, because experimentally validating protein antigen candidates is very time and resource consuming. And so it would be great if we had a computational tool that could be used to just screen potential candidates and then predict whether this is a good antigen candidate or not. And for this, we really require high quality data for training such a model. And PlasmoFAB provides manually created Plasmodium falciparum protein data for the training of these computational pre-screening methods. And here's an overview of how we created PlasmoFAB. You can see different stages here, and I will walk you through those individual stages one by one. So PlasmoFAB consists of manually created proteins that were included based on a combination of prior knowledge, established computational models, domain expertise, and literature search. And in the end, we had 438 protein antigen candidates. So this is our positive set in our machine learning model in the end. And then also the 384 intracellular proteins, which are considered negative examples in our training of our machine learning model in the end. And we start from PlasmoDB. So PlasmoDB contains sequences for those different Plasmodium variants. And so, for example, also for Plasmodium falciparum. And then what you can do is to look for known antigens. And known antigens you can find by looking at data from IEDB. So IEDB is a database that contains information about immune system processes, so antigens, again, for T-cell antigens, also peptide binding for MHC alleles, for example. But here in this case, we were interested in these antigen candidates for B-cell produced antibodies. And so we were looking for exact matches or high or medium confidence blast hits here for our sequences in that IEDB epitope database. And if there was an antigen present, then we included that protein into the positive set. Then we know that for the majority of Plasmodium falciparum proteins that are exported into extracellular space or are integrated into the cell membrane, we have those PEXL or HT motifs present. And so we could also look for those motifs by just looking at the annotation from PlasmoDB. And so we could include those there then easily, because those are the ones, if they are at the extracellular space or at the membrane, then those proteins can stimulate an immune response and can be bound by those antibodies. Then we had an additional data source. So this is for the sporozoite surface exposed proteins. And there was a recent study by Swearingen et al. And in that study, they validated experimentally proteins that were found on the membrane during the sporozoite phase. And they had different confidence levels there. So the highest confidence level was 1, and the lowest confidence level was 6. So we included those proteins with confidence level 1 to 3. Now, there are also varying surface antigen families, and there are also other known membrane proteins, and we also try to include all of those into our positive training set. So proteins belonging to the VSA families are generally located on protein membranes, so we could include those. And out of those, we had the PFEMP1, Rifin, and also Stevor here. Then additionally, there is a lot of knowledge about proteins which are at the membrane level. And so we could also include those which are known from scientific literature. If you want to build a high-quality data set for training a supervised machine learning model, you also need negative examples. And so we also created a set of proteins which are not bound by antibodies in those people who are infected with malaria. And those are, for example, intracellular proteins, because in intact cells, those will never be seen by antibodies which are floating around only on the outside of the cells. And we identified those by domain expertise and also literature search. So more details you can find in the manuscript. Now, we wanted to check when we had our data set directly whether available prediction services were already good enough to predict whether a protein would be in the positive or in the negative set. And so available pre-trained method that existed there were services that predicted whether, for example, a protein is a transmembrane protein, so contains, for example, transmembrane helices, or has a signal peptide, or there are also tools that let you predict the location of the expression of a certain protein, and those will then give you the location. So this could be, for example, also Golgi or some other location, but also whether it's exposed at the surface or whether it's at the cell membrane. And this could also be predicted with those tools and then checked against our benchmark. Unfortunately, the performance on PlasmoFAB was not really satisfactory, so the MCC was smaller than 0.5. So we had to develop something else to really get to a prediction method which could be used for this pre-screening that I already talked about. And normally when you build a machine learning model, you want to have a strong baseline that you compare to, such that you know that you really developed something good when you can beat those baselines. And so one of the baselines that we implemented was a support vector machine with a string kernel, and here in this case, the oligo kernel. And the oligo kernel considers words of a certain length. So those words omega of a length k. So here in this example, you would have length 1, and you would only look for amino acids, basically. If it's length 1, then each amino acid is a different word, and then you can look at these curves here to see where those signals really occur. So for example, if you look at the Q here, then you see that it occurs here, here, and also here, and so you have that signal. And you can even smear that signal a little bit by controlling for the wideness of these Gaussians here. We didn't just want to use those baseline methods, but we also wanted to develop something that is state-of-the-art. So we looked for language models which could give us an informative embedding of our sequences. So you heard all about these language models, and basically here the implementation is that each amino acid is considered a word or a token here. And then when you train these models with self-supervision, then basically what you do is you mask certain words, so in this case, the amino acids, and then you try to predict those amino acids from the other amino acids, which are not masked. And through this, and through seeing many, many, many protein sequences, you can train an encoder here, so a transformer basically with self-attention, that in the end gives you a very good embedding of your sequence. And this embedding is then a very high-dimensional representation of your amino acids, still containing the position information. And what you can then do is just take that representation, so that's embedding together with the position information, and train a convolutional neural network, for example, but you can also train another supervised learning algorithm here. So in this scenario here, you would still have that position information. So if you have a longer protein, then you would also have a longer representation here. If you want to have something which is independent of the size of the protein, then you can also do something like mean pooling here, and then get a representation that you can use no matter how long your protein is. But in our approach, we were interested in the first one here, so basically having the position information here, still having that good embedding here, and then running this together with an SVM or logistic regression with our label of interest, which was then this antigen candidate prediction problem. And so we evaluated those embeddings together with support vector machines and logistic regression. And we basically compared two of the best protein embeddings here, ProtT5 and ESM1B. And we compared these methods to the SVM with the string kernel and also those pre-trained prediction services. And for this, we basically created a benchmark from our PlasmoFAB dataset such that we could compare also to our method that we trained on part of that data in a fair way. And so for this, we created a test set which had little homology to the training sequences. So basically, we chose those sequences for the test set, and none of the training sequences had more than 30% homology to any sequence in the test set. So the homology was even smaller than 30% between any sequence of the test set and any sequence of the training set. And this was ensured by MM62. Now, when you look at the results here on the right, you can already see the winner here, but what you can see is that this embedding here from those language models really helped to boost the performance considerably. So those pre-trained models, for which you can see the performance here at the bottom, were much worse regarding MCC compared to those methods based on those embeddings from those self-supervised language models. And combining feature embedding ProtT5 with those linear regression method here led to the best performance. And what you can also see is that that very simple interpretable model here contained better performance than some of those pre-trained models here, even though it's a very interpretable model and should therefore probably be preferred over at least these three here for this prediction problem, antigen candidate prediction, for proteins of Plasmodium falciparum. So those results clearly indicate the importance of available high-quality data for model training, and also that it was very valuable to create this high-performance data set. Okay, so to conclude, we created PlasmoFAB, which offers Plasmodium falciparum-specific protein sequences with high-quality labels and contains labels that distinguish between protein antigen candidate and intracellular proteins. It provides a predefined test set to allow easy and unbiased comparison between different pre- and self-trained prediction models. And PlasmoFAB allows to train prediction models for computational pre-screening of Plasmodium falciparum protein antigen candidates. If you're interested in the dataset, you can find it on Zenodo by following the link from this QR code on the top right. And if you're interested in our source code, then you can also find it on GitHub by following that QR code on the bottom right. And if you're interested in this topic area, this intersection between machine learning, computational biology with infection research, feel free to reach out to me at the conference and or via email if you're interested in any collaboration or position as a PhD or postdoc in the future. So we would be very happy to talk to you about any opportunities there. So thank you very much for listening to the talk and your interest, and I hope to see and communicate with you at some point. Have a nice day.